Hey friends, good day to all. This is the continuation of the fertilization part. In the previous part, we have discussed in detail about the concept of fertilization. Please watch that, only then you can understand this part. I have put in the I button, so go and watch. My dear friends, as we have been studying, the sperms are being inseminated and after that they are being transferred to the fallopian tube at the site of the fertilization, as well as from here the ovum are also getting transferred, and we have learned all these process under the approximation of gametes in the previous class. So after the approximation, even though the both the gametes have came at the site of the fertilization, there are some difficulties, there are some barriers that have to get overcome in order to get fertilized. So if you see this image, I have put the image of the ovum from here. This is the barrier, that is, this is the actual nucleus, female pronucleus, that is going to participate in the fertilization process, and the sperm is outside. And so in order to enter the sperm nucleus into the ovum, it has to overcome these two barriers. One is the corona radiata and another is the zona pellucida. So this is the zona pellucida and outside is the corona radiata. I will show by enlarging this. So here you can see very clearly that this is the corona radiata and this blue one is the zona pellucida. So these two barriers have to be overcome in order to get fertilized. Okay, so let us study in detail. So my dear friends, the first thing that is taking place after the approximation of the gametes is the capacitation of sperms. What is the process of capacitation? It is the process of maturation of spermatozoa, under which the glycoprotein coat that is present at the head is getting removed to increase the motility. So capacitation is the process of the maturation of the spermatozoa. This is helping in the contact and fusion, so that is why we have included here. And after this, what is the major process, that is the penetration. So that penetration, the penetration process, that is studied under 4 phases. Okay, that is studied under the four phases. First is the acrosome. There are some enzymes that is released. Those enzymes will disintegrate the zona pellucida and the corona radiata, and those enzymes, the process of releasing of those enzymes is known as the acrosomal reaction. In this acrosomal reaction, what is taking place? A release of enzymes, and this process is the first phase. And what are those enzymes? One is the hyaluronidase and second is the acrosin enzyme. This is the proteolytic enzyme. Whereas, and this what is the effect is studied under second phase, that is the disintegration of barrier. This is the second phase, and in this second phase you can appreciate the disintegration. So what is the function of the hyaluronidase? It is also known as a corona penetrating enzyme, because it will dissolve the hyaluronic acid in the ground substance of follicle, that is the corona radiata. So corona radiata. And the acrosin enzyme is dissolving the zona pellucida. It is acting on the zona pellucida. So we have learned here, this is the corona radiata and this is the zona pellucida, and corona penetrating enzyme is the hyaluronidase, and next layer is the zona pellucida, that is being dissolved by the acrosin enzyme, and all these are released from the acrosome of the sperm. You can better appreciate in the structure of the spermatozoa. Okay, and it will facilitate the penetration of sperm. So next, Before going to the next stage, let us question ourselves that why only one of many millions of sperm successfully fertilize? Why only one is successfully fertilizing the ovum? And that is meant by how is polyspermy prevented? One is the only 1% of the total that are inseminated, they are reaching the uterine cervix, and only 300 to 400 from there they are reaching to the fallopian tube. And even in the fallopian tube, from among the 300 to 500 sperm, only one is getting fertilized. What is the reason? What is the reason? Okay, so the answer can be understood here. The answer is found in the next step, that is, after the barriers have been disintegrated, there is occurring the fusion of the sperm with the plasma membrane of the ovum, that is known as a vitelline membrane. So here you can appreciate, see, this is the zona pellucida, na? So this is the plasma membrane of, that is the vitelline membrane. So the fusion of sperm with the vitelline membrane inducing, that is triggering, that is inducing a wave. That wave is the calcium wave, and this calcium wave is the, it depolarizes the vitelline membrane. And there are three effects of this wave. One, it will release the cortical granules. You can appreciate here in this. So here there are the granules, and it will release the cortical granules, and these cortical granules, after the degranulation, they will hydrolyze the Zp protein, zona pellucida protein receptors. Here are the zona pellucida protein receptor. With this, it is attaching and releasing the its enzymes, what is known as the acrosome enzyme. Okay, so if the Zp3 protein receptors are being disintegrated or hydrolyzed, then the other sperms cannot attach here, and so it is being prevented. This is one process. Another is, by the process of this degranulation, the vitelline membrane alters in such a way that it blocks the entry of further sperms. And this is the second way. By these two waves, the polyspermy is being prevented. So this degranulation is leading to one and second. First one is hydrolysis of Zp3 receptors, and due to this, what is happening? The prevent of binding of more sperms, so the next sperms cannot bind, and so the polyspermy. And second process is, it alters the vitelline membrane in such a way that it blocks the membrane, so that it prevent polyspermy. So in this way, the combined effect is taking place and leading to the preventing of the polyspermy, and inducing of the calcium wave is the third phase. And what is the fourth phase? The fourth phase is syngamy. And how that is taking place? Syngamy, this inducing of the calcium wave, this has another effect as well. It is releasing the cortical granules, as well as it causes the completion of meiosis 2 that is being arrested. So now that arrested cell got bail, and now it can move further, and it will release the ovum. Second meiosis is completed now. There is no more arrest, and that is being triggered and induced by the calcium wave only, and the second polar body is formed. So as a result of this calcium wave, and this meiosis 2 is completed, and from ovum the sperm gets fertilized, and the process called syngamy takes place. Syngamy means the fusion of gametes. Syng means fusion, and gamy means gamete. So the syngamy takes place, and the formation of Synkaryon means the fusion of nucleus, and that synkaryon is the zygote nucleus, and thus the zygote is formed. This syngamy is the fourth phase. As we have told, there are four phases under the heading of contact and the fusion of gametes. So please do subscribe and share the video with your friends who are bioscience students. Never forget to like the video and have a nice day. Until then, goodbye.